finding data online. Basic idea is that we will use public data as much as we can. We need data. Fortunately, there is a lot of data. I found that there is something called the International Nucleotide Sequence Database Collaboration. This is a link, we can open it. Let's click it here. Let's see what happened. International Nucleotide Sequence Database, it is long-standing foundation that operated between DDBJ, well, I'm going to give this keyword, DDBJ, MBL, and NCBI. So this is connection between three very large data repositories, literally the National Center of Biotechnology Information, and they think called bioinformatics, to be honest. They call it biotechnology information, because when they created this center, kind of bioinformatics was still something very new. This is part of a big institution called the National Library of Medicine in US and in the National Institute of Health. So just to give you an idea, the National Institute of Health is, a very American thing. It's a very big campus. It kind of, it's bigger than any university have seen in Turkey. Maybe Koç is kind of the area, but it's flat. Koç has a lot of mountains. So it's a big campus, and they have different institutes. They have the National Cancer Institute and the National, I don't know. I know the Cancer Institute, they have, for every kind of disease, they have a center. And they have, in particular, a branch called the National Library of Medicine. And this is a very old institution from the 19th century or even before maybe. And they collected all literature about medicine. And what surprises me, they are in a building that is bomb-proof. They designed that in case of an atomic bomb, the library will collapse, everybody will die, but the books will be saved. So when they survive, the survivor of the atomic bomb will be able to get all the books to survive. Well, that's the American version, is the one that I know. Then there is the European Bioinformatics Institute, which is part of the European Molecular Biology Laboratory. That is something that you should be aware. There is an institution called MBL. They are mainly based in Germany. Kind of the homework is in Heidelberg, which is one of the most beautiful cities in Germany, in my opinion. And but the bioinformatics part, they do it in Cambridge, in UK. And well, I have a friend working there, and they kind of like it. It's complicated now with all the Brexit. They don't know what is going to happen, but they think they are going to be fine. And finally, there is a counterpart in Japan, the DNA Database of Japan, which I don't know really. These three people are essentially doing the same thing. So they coordinated to have the same data. It kind of doesn't make sense that you do the same effort 3 times and then you will have one data in one data in another. So the first thing they do is that every night or with some other protocol, they share everything. So if you put your data in the Japanese database, in 24 hours, it will be in the European, in the American database. Okay, so you can be pretty much sure that the genomic data will be in all of them. Then they do some other processing. So they are not identical. They are not mirrors of each other. The data, the genomic data, the raw genomic data is the same, but they process, and the processed data, the curated data, or the derivative data is different. So it is good to know what each of one does. So I can tell you immediately that my bias is the NCBI approach, because it's the tool that I use. I was under the American influence, and I've been a couple of times in NCBI. And I even went to a course, I learned some stuff directly from the people there. And then, but the European is super interesting, and I would like to know more. So one of the goals of this course will be that you teach me how to use the European database. I'm going to show you what I know about the, maybe it will be like some of you studied the American some of you study the European, and some of you study the Japanese, and to see what is interesting here, what is different, what we can do, we cannot do. So we are at 10 people, we can do 10 times better. The other important thing, beyond that all the data is free, is that according to, at least to American law and probably European law, if your project is paid with public money, then your data is public. I mean, taxpayers already paid for your work, so your work should be available for everybody. That is, by the way, the reason why I think all my courses should be online, because I'm paid by public money, therefore everybody is the owner of this information. So, if your project was paid with public money, you are forced by law to put your data, at least in this country, the people that pay for this, they say, okay, if you give you money for this project, you have to put your data in a public database. Then the good journals, let's say Nature and the good ones with big impact, don't publish you unless you upload your data. Somehow it doesn't happen in all areas of molecular biology. I don't know why, but in my opinion, it's going to be more and more important. For what we say, for genomic data, this is the rule. For any sequence data, even gene expression, you have to upload your data before publishing. So what people do is they upload with a code that say this is private, and they give access only to the referee, to the reviewers. And after the paper is approved, then it becomes public. So for example, maybe I told you, I was involved in a project about the salmon genome. So in my former lab in Chile, we get a partnership with Canada and Norway to sequence the genome of the salmon, because, you know, Norway, Canada, and Chile are the biggest salmon producers in the world. Because Chile has essentially the same climate that Norway, but it's in the opposite side. When it's winter in Norway, it's summer in Chile, and when it's summer in Norway, it's winter in Chile. So they can compensate the production. And the salmon genome was an interesting thing, and we found some public money and we paid for it. And the condition was that we got the genome and we have it for 6 months. There is a condition called embargo that allow us 6 months of advantage to analyze the data. After 6 months, everybody got the data. So it will happen probably in most of the project that you will be involved with, that your data will be for you only for a limited time, and after that everybody got access to your data. On the other side, the advantage is that you will also have access to other people's data, so it is fair. Okay, so I'm going to show today the NCBI website. The goal of today is to get a general overview of the thing that we can do in the NCBI website. I'm going to use it and see what kind of files we can get, at least a basic level. And that will allow me to explain, to give context of what is going to be the next class. I insist, my bias is NCBI because of the tool that I know. I'm not saying that it's better, and it probably is not. Probably they have their own advantage and their own weakness. So try to look at the other tools, the other databases also. The first distinction that I want to make here is the difference between Genbank and Refseq. Genbank is the origin of this website, is a database of all sequences. So if you do an experiment, you go to a web page and you can upload directly. You don't need to ask permission to anybody, you just upload. That means the advantage is that you get most of the data very quickly. The disadvantage if you don't know if this data is correct or not. I mean, maybe the labels are wrong, maybe the sequence is labeled like one organism, but it's contamination, and there is no quality control here. So there is a second level called Refseq, the reference sequences, that is a subset of Genbank that is curated in the sense that it is cleaned by some humans. So Genbank, if you like, is automatic, and Refseq is human processed. So people clean it. There is some quality control in the Refseq. Then DNA databases, there are two that I want to focus here. There are more, in fact, but let's start with this. There is a first, and you are going to see the Genbank nucleotide, but also the Refseq nucleotide. This separation between Genbank and Refseq is going to appear also in every other case, in every, well, at least in the sequences. Okay, there will be nucleotide Genbank and nucleotide Refseq. And in protein, we have protein Genbank and protein Refseq. We have a database called Nucleotide that essentially put everything. Now, these tend to be short. The nucleotide is not containing the full genomes. It's more like you sequence a fragment, a gene or a 16S or something like that. Then you upload that fragment, okay? It's not for full genomes. There is another database, the sequence record archive, sequence read. This is super interesting. Here you can download all the output of the new generation sequencing. In fact, I should have mentioned there is a database of sequences that, I mean, the output of the sequencing from the old technology is called the trace database, and the SRA is the sequence of the new generation sequencing. So if we want to do sequencing for this course, we can download public data. Someone else sequence it and you can get that. And also you get the idea that if one day you do a project and you sequence your own data, you will also have access to publish everything. Protein is very much like we say the nucleotide. Many of these proteins sequences are automatic translation, in the sense that many of the proteins that we know them in theory. Let's say this. You extract RNA that you identify as a messenger RNA. You sequence it. When you get the RNA sequence, it is very easy to predict what is going to be the protein. You know that there is a rule, the codons and so on. So you know the protein sequence. That doesn't mean that you have seen the sequence. It does only mean that you have seen the gene expressing that protein. So in general, are hypothetical proteins. We not necessarily have seen them. But in this case, they also include data from other databases that, hint, hint, we are going to take a look. There's something called the protein database, protein data bank. There's something called SwissProt, which is a Swiss project of proteins. But there are other databases focused especially in proteins. These databases tend to have the 3D structure. Okay, these are databases that contain real protein that people have identified. Then there is another group that is going to be important for us, the kind of protein clusters. There is also named the cluster of orthologous. They started essentially with bacteria, but now they are also including some eukarya and mitochondrial and so. The basic idea is they try to identify when you see the same protein, quotation marks, the same protein in different organisms. Like let's say RNA polymerase. RNA polymerase is a protein that have a specific role, and for doing that, they tend to have the same sequence, not exactly the same, but a very similar sequence in different organisms. So you will have, I don't know, 100 different prokaryotes, and each one will have a protein, and the protein are going to be very similar. So you put them together and you call all the members of this, you call the cluster, and you can assign a function to all protein in this cluster. Why we care about this? Because when you find a new sequence, you find a new bacteria, let's say, and you find the genes, you can say, okay, this gene, what is this gene doing? If you can find that this gene matches one of the clusters, then you can at least hypothetically, you can say, okay, this gene is doing the same as the other genes in this cluster. Okay, so what I mean is this: if you find an animal that you've never seen before, but it moves like a duck and swims like a dog and speaks like a duck, then you say, okay, this is duck. If it seems to be something, you tend to assign the same name. Okay, I want to give you already an example that I use every time. Bioinformatics is very much like when Europeans first went to Australia. You know, Europeans knew the animals in Europe and Asia and in Africa. But when they get to Australia, which is a continent that was isolated for a longer time, they start finding animals that never seen before. So how you understand things that you have never seen before, it's a complicated problem. So how you understand a kangaroo, for example, if you've never seen a kangaroo? Well, that is essentially what we're going to have here. Just keep in mind that we know probably less than 1% of all bacteria that exist in the world. We really don't know anything about bacteria. Or in general, yeah, any microorganism, we know by name like 5% of the bacteria, and by sequence, less than 1% of all bacteria. So if you take a sample, even you do like this, you take and you sequence this thing, you will find many things that you've never seen before, nobody's seen before. And you will try to make sense of that. So that is the big challenge, how to understand things that you have not seen before. Okay, what else? In the case of protein database, the protein, we have also a couple of things that allow us to understand the protein. One is conserved domains. You probably know that the proteins are that have a structure beyond just a set of amino acids. You have, you know, alpha helix and beta sheets and things like that. And you have higher level structure, like there is something called zinc fingers and pattern that allow the protein to bind to some domains or to some DNA or some binding site. This thing, if they have a role, tend to be conserved. So there is a database of domains that, like, part of protein that have been observed in different proteins that do different things, but they share some part. So this domain eventually can get identified like a binding site or something like that, or active site, catalytic site. What is the name? The active site, no? This tend to be conserved, and it allows you, in some cases, it helps you to understand what is the protein doing. Then we have this database of structure in the case that nuclear magnetic resonance or X-ray allows to identify the structure of protein. Then they are stored in a database that we can access. Let's move to gene expression. I very much like the gene expression database, and it is called Gene Expression Omnibus. Omnibus here is not only the bus that you take. Omnibus literally means it carries everything. Bus is what is Latin for everything? Omnibus is not the bus that we take to move. There are several parts, but here I am speaking about two things. One is the data set that essentially, when people do an experiment, you get, let's think a microarray. A microarray has, I don't know, 10,000 genes, and you measure the concentration of each one of the 10,000 in a given condition. So that will give you one sample. A data set is a series of samples taken with the same platform, with the same slide. So you essentially see the same cell in different conditions. You see how the gene expression change to different conditions. The profile is the horizontal version. You take one gene and you follow the same gene through different conditions. So data sets are vertical and profiles are horizontal. Okay, you have genes in the rows and conditions in the columns. Data sets are the columns, profiles are the rows. And then there is a database of probes when people say, for example, if I did qPCR or I did a microarray, what are the probes that I use? The oligos, the primers that were used for doing that experiment. That can save you a lot of time because at least you know that someone else did this experiment and it worked for them. So we have to check that. You can save a lot of time. Then a database that is super important for me, and this is going to be the subject of next class, the taxonomy database. Everybody know what is a taxonomy, no?
Yes, we know.
You know what is taxonomy? Good.
I know.
Okay, the question that I will give you, what is the difference between taxonomy and phylogeny? That is a question for next week. Okay, but we're going to explore the particular way of this database. And then there is something else that is kind of specific, but will be good. It's called the medical subject headings. And the keyword here is control the vocabulary. This is an important idea that I want to stop a minute to discuss. You will find that different people use different names from the same thing. So, for example, some people say yeast, some people say Saccharomyces cerevisiae, some people say S. cerevisiae. So if you are going to look for a yeast and you use the wrong word, you will not find it. One of the things that researchers have found that is a good idea is to have a control of the vocabulary, meaning that you cannot use all words. You have a limited set of words and you can only use words in this vocabulary. You have, I don't know, 1,000 words, 10,000 words, but not more than that. So if you are going to call it yeast, you cannot call it yeast. You have to call it Saccharomyces cerevisiae. You have to be explicit. It is kind of annoying when you have to find it, when you have to store the data, but it's super good when you're going to find the data. The idea of control the vocabulary is super, super important. It's one of the ideas that we want to emphasize here, that the same object should have always the same name. If you get something from today's class, it is one object, one name. So you cannot get confused with the name. You use one name and you get to the same object always. Control the vocabulary are a strategy to achieve that goal. The same object with always the same name. So if you know how to use mesh, it will help you to search better in the database. These two tools are important for finding data. Then a couple of things that we should say. One of the important things that they do well, remember that this is part of the National Library of Medicine. So they have the index of every paper published in every journal connected to biology or medicine. Including, that is interesting because some journals like Nature have a lot of biology, but they also have other areas and they also have them. So if you want to find the title, the author, sometimes the abstract, everything is in Pubmed. So usually when we look for a paper, we start with Pubmed. Now, for different reasons, not all papers are public. Some papers, the journals ask you to pay money to get access to the journal. So Pubmed has all the references, the citation and abstract, but not the content. For some of them, they have in a separate database called Pubmed Central, and they have a text, full text of the paper. How they can do that? Well, some journals are public. For many, many papers, not all, but a lot of paper, they will be available here. If you, depending on the journal, you can republish your data. You know, the different journals have different policies about who owns the copyright. If the journal allows you to keep the copyright, like an author, you can submit your text directly to PubMed Central. Some journals are public. Some journals are public after some time, like 1 year old data is public. So there is a good thing. And if no, in some cases you get the link to the original paper. In fact, I don't know, did I send you a link? No, I sent in the other course. I will show you later. There is a paper about learning online that we will probably read it for learning online.
Do you think it's safe to break the paywall by some websites or some tools, maybe?
What do you mean?
I mean, there are some papers that are not public. So, do you think it's safe to break the paywall by Sci-Hub, let's say?
Well, the paywall. No, NCBI is, they are very careful about this copyright because they are a part of the business, no? So all these things are outside of the paywall. Sometimes they have link. We will take an example, but sometimes they have a link to the website. If there is a paywall, they point to the paywall. Now you should know that in the university we have access to some journals that are beyond the paywall, and there is a way that from home you can connect through the university with a proxy that allow you to access the papers that are beyond the paywall.
Yeah, I recently discovered that, but I heard that they don't give the authors the money they take. So
I know, sure. No, that is, but that is a problem. That's a longer story. Like, I mean, one of the hardest thing in science today is that we are paid usually by taxpayers or by a private university to do our work, and we publish it, and we don't gain any money. When you publish a paper, you don't get paid anything. Moreover, to publish, you have to go to the journal, and the journal has to send to a reviewer. I am a reviewer for some journals, and it takes a lot of time, and I'm also not paid. So essentially, the university is paying me to write papers and is paying me to review papers, and the university doesn't get paid, I don't get paid, but the publisher get paid. And
that's not fair, is it? Like,
it's not fair, but they have the control it. It's a very controversial thing. On the other side, the problem is this. Traditionally, when papers are based on paper, publishing was expensive, so the publisher had really to invest money. And that investment means this, that you are only going to publish a paper if the paper is good, because you cannot risk to publish bad papers. If your paper has bad reputation, nobody will buy your journal. If your journal is publishing bad papers, you don't get money. So traditionally, journals were very careful to select good papers, and they give you some kind of guarantee of quality. Okay?
Mm-hmm.
But today, since most of the money is not coming from the readers, let's say this. Today, you don't pay to read the paper, you pay to publish the paper. In fact, yeah, it's even worse. If I want to publish a paper, many journals ask me to pay, and it's $1,000, $2,000 that I have to pay to publish my paper. So the journal have a lot of incentive to publish a lot of paper. The more paper you publish, the more money they get. The problem in that case is that you don't know if the quality is good or not, because essentially the journal has all the reasons to publish everything. And in the best case, even if everything is good quality, you have many, many papers. So in all times, every journal has, I don't know, 20 papers, and now they have 80 papers. So finding good papers is going to be more and more difficult. That is why we need this kind of databases. We need to understand how to navigate these databases, because otherwise it's very easy to get lost. By the way, people say that half of the papers published are never read by anybody. And you can essentially see that if a paper is good, they will get citation by someone else. Many papers don't get any citation, or they get cited by the same author. So that doesn't count. So it is super important to see. I think I have only one paper with good citations.
Sometimes really good papers don't get citations for a long time before they discover to be like.
Yeah, that happens. I mean, there is a lot of chance in this thing, no? There is a lot of, there is even a law, you know, in mathematics, you know, the Pythagoras theorem and the, I don't know, the Newton rule, and there are many things that have the name of the people who discovered it. Well, the truth is they didn't discover it. Most of the results in mathematics have the name of someone else. And the same will happen here, that people who do the research are not the people who are going to be recognized. So there is a lot of chance in this. But for now, you have to know how to find the papers. We need to know how to navigate them. And later we find how to write good papers.
Okay, let's proceed then. Thank you.
Yeah, no, I like a question, so no problem. I like questions, so let's keep. I like answer better anyway. Okay, we're back. Literature. Something that I like very much is that they have, the NCBI has a set of books that are very, very expensive, and they're available online with some restriction. For example, some of them you can search into them and read the article, but you cannot browse them. You cannot go page by page. Some of them you can read page by page, but you cannot download them or print them. So, well, in some cases, it is super useful because we're speaking about expensive books. Well, there is, of course, the search and all these other items. Okay, a couple of more comments here. There is some genome database. There are some subtle things that I will not enter in detail here, but there is a point that there is a difference between the genome and the assembly, because let's say the human genome has several versions. You probably know that since the Human Genome Project finished, there have been several versions. So because the DNA assembly problem is a complex problem and you have several solutions. So sometimes you are not very sure of where all the parts are fitting. So you may have more than one assembly from the same genome. That is why there is a separate database for assembly. There is some maybe old databases that expresses sequence tag that is EST is kind of what people do when you don't have money. If you don't have money to sequence an eukaryote, normally you have money for sequencing the messengers. So the ESTs are essentially, you take the mature RNA, you copy to cDNA, and you sequence that. So you get an idea of not all the genome, but the genes. Okay. Maybe today it's cheaper, but this was a very common technology 5 years ago. Genome survey is also when you don't have full. Now that people have better sequencing technology, these things are a little more obsolete. Well, you can read this later. Okay, this is unfortunately this got cut here and should not have been shown, but there is some database of the projects that are being carried on. The project here is more like who is doing what are the companies. I see what happened. I made a mistake here. Anyway, what are the institution? What are they trying to do? Who is paying for things? Then sample means essentially the physical thing. When you sequence something, you can have one molecule and sequence it several times. So at some moment you should connect this DNA sequence with the molecule. The biosample is a description of the molecule or the I don't know, pipette or the Eppendorf that got the thing. So for example, if you want to resequence something, you should be able to find the original sample. Okay, so there are databases for all these things and many other things that are beyond what we can do. So my suggestion is now we take a look at this and we can all talk.